So we already talked about a little bit about those discriminant analysis. So here we try to discriminate between two systems. So either we are looking for probability of something happening versus not happening, and then we take these probabilities and we take log. We convert the results into a log scale, since the probability are small numbers, and those their divisions with those small numbers can give further small numbers. So we can use that log scale in order to pick them in the whole scale. So this kind of analysis is where we apply those discrimination or discriminate analysis. So we use that in order to do our promoter predictions, while taking the examples from one of the the best quoted example is CPG islands, identification of CPG islands. So we'll talk about that in this section. We know that Markov chains can be used to discriminate between two options by calculating a likelihood ratio. In fact, we'll calculate a log likelihood ratio. What are CPG islands? So CPG islands are present in a genome, and they indicate the presence of genes. So we see that whenever C and G they are present together, the C gets most of the time in the genes in the genome, it gets converted into T's. So we'll observe more T's and C's. So, but this process of converting this C into T is reduced in around the genes or in around the coding region. So wherever we can get those CPG, we can expect that there is some gene coming in that region. So given the sequence from CPG islands and sequences from other regions, so we take the sequences from those intergenic, or you can say around the non-coding regions, and same way we can take some regions which are coming from the genes. We can look into the frequency of these C's followed by G's in those both of them, and then in the end we can compare them, and we can estimate whether we can find a gene or we cannot. So a model we make over here is for CPG islands. We call it as a plus model, and we can also have a alternative or a null model to compare, where we don't where we get the sequences coming from non-CPG regions. So that is a negative model. We calculate the parameters for both of them. What we do here is we are getting some data in this example, which actually I got it from Durbin's book, which is one of our textbooks. So what they did is that they took human sequences containing 48 CPG islands. So the regions from where those CPGs are enriched are present, we call them as CPG islands. So remember, there is a whole ocean and there is a small piece of land within that ocean, so that is called as an island. So same way, the whole genome is really big, but there are some places where see those CPGs concentrated, so we call them as CPG islands. So they take 48 CPG islands from 60,000 nucleotides long sequences, and they make those transition probabilities. A represent here as a transition probability, and S is a nucleotide S, which is any nucleotide, and T is the second one. So S to T, moving from S to T, is the transition probability. So we use these transition probabilities in this example, and in this equation we have C number of times this T follows S, and we have K, which are actually the states which are there in this system. C plus ST is actually the number of times S is followed by T. Plus means while we are within the CPG islands. Same way we have C minus ST, where without CPG islands, or which are the regions CPG minus regions. So for that we have the transition probabilities calculated like this, and we construct the tables, these transition probability tables, both for CPG islands and non-CPG islands. When we put them together, you observe that in these matrices, the top part is where is the second nucleotide, and this one is our first nucleotide. So what we are seeing here is how many times this A is followed by A, C, G, and T. Same way, how many times C is followed by G in this plus region? So we observe that it is 0.27. As compared to small, but still it's a big number when you compare with the control, which is the non-coding region. So here you see that it's just 0.08, it's just 8%. So 27% versus 8% of occurrences is a big number. So let's see, when we calculate, when we put that in our equation or in our discriminate analysis, what do we get? And same is for rest of these nucleotides, like how many times G is followed by A, C, G, and T, and rest of them. So we calculate the log odd ratios for the sequence by log of 2. We take log 2. In log 2, what happens is that the numbers are converted such that if you are getting a number 2 in log 2 scale, that is actually 4, which is actually 2 raised to power 2. So we are taking those numbers into those powers of 2 actually. So we take those probability of seeing that X in CPG model versus X in null model. So we take, for example, a probability of G following a C in this top model versus negative model, and whatever answer we get, we convert it into log 2. So when we do that, we can present it into more ugly form like these equations. So we can present it in a generic format where we can have A plus X I minus 1 X of I. Here actually we are modeling it for any nucleotide. So this is another form which was observed in the book. Same way, AX plus and AX minus are also substituted with something which they label it as B or beta, and we get a table for this. So you don't need to worry about much as far as these ugly equations are concerned. Just try to grab the general concept. So when we get those beta values, we can also call it as bits or binary digits. So these bits are actually, since we are getting on a log 2 scales, so they use these words like bits, etc. So that's perfectly all right. So we will get some numbers, and in this case, if you observe the beta value for the change in which we were interested in, so that is 1.812. How many times we observe a CPG? How many times G follows a C? Remember guys, this is CPG. It's written in order to tell you that both of them are in one strand, rather than thinking that they are something like this. No. So this CPG here is observed, the value is really high. So that's what is now we get this after this discriminate analysis, we can see that presence of this CPG in coding regions or in the regions where we have genes is higher as compared to the negative regions. So this is how we can use Markov chains in order to do this discrimination or discriminate between coding versus non-coding regions with respect to presence of these CPG islands. So for different sequences X, we can use those beta scores, and we can compare them. And since different sequences are having different lengths, so we can get those beta scores and divide them by their lengths, so in order to have a fair treatment for a short sequence versus a long treatment. In our discriminate analysis, if the score is negative, then we say that the sequence is not in a CPG island regions. If the score is positive, then obviously it is. And how we get those negative and positive numbers? Remember guys, we are dividing two numbers. So we are getting the frequency in plus versus minus, and that these numbers we are getting in actually when we divide them, and when we get those numbers less than 1, and when we convert it into log scale, it gives us a negative value or a negative number. So if the answer is greater than 1, means the top part in this equation or in this division is higher than the lower part, so our answer will be positive. So in this plot, we see that we have some CPG rich sequences and we have some random sequences. So when we compare them, the bit scores are higher, and as far as those these CPG rich regions, and bit scores are lower, our negative numbers are where we have those random sequences. So we hope that this technique works. So Markov chains can be used to discriminate between two options, and we can calculate their likelihood ratio, and we can compare them.